Hi Year 12s. Today we're going to be looking at the cell membrane. So we're going to be looking at what makes up the cell membrane, how different materials are transported to and from the cell across the cell membrane, and what are the properties of those molecules that need to be transported across the membrane look like. So this will be 3 videos for this topic. The first one, the one we're looking at now, is just the structure of the cell membrane itself. The second video will be looking at how transport occurs across the cell membrane, and the third video will be looking at the different properties of the molecules that need to be transported across the membrane. So let's begin. As per usual, we've got our need to know here. So the need to know is basically just the SACE curriculum guidelines for this video and for the next 2 videos. So this will only be in this video. If you want to take down any of these, I recommend you pause the video, write them down, but this is the content you need to know from these next 3 videos. First off though, let's start talking about tissues. So when cells of the same type all come together and they perform a specific function, that's when scientists start to classify them as tissue. So some common types of tissues in animals include epithelial tissue, connective tissue, nerve tissue, and muscle tissue. At the moment, there's a very controversial discussion across the scientific community on whether or not we accept bone and blood as a type of tissue, or do we say they're not types of tissue. So that's one of the ones that's debated heavily at the moment. The actual cell membrane though. So the purpose of the cell membrane is to basically separate the insides of the cell from the outside of the cell. So the intracellular environment needs to be different from the extracellular environment, because we need inside our cell very specific requirements in order for all of our metabolism, etc., to actually take place. Every cell that we have found contains a cell membrane, and that cell membrane is used to control what enters and exits the cell. It also plays a part in cell communication, cell adhesion, as well as the binding of hormones. But what actually makes up a cell membrane? Well, every cell membrane is made up of a phospholipid bilayer, which we'll look at on our next slide. There's 2 different types of proteins. We have integral proteins. Integral proteins are proteins that are embedded inside the membrane and physically can't move, so they're permanently in those locations. There's also peripheral proteins, which are not fully embedded into the membrane and can actually move back and forth in different positions, so they're not in a fixed location. In animal cells, you'll find cholesterol, and cholesterol helps prevent the cell from becoming too fluid or not fluid enough. We also have glycoproteins, which are just proteins with carbohydrate chains attached, as well as glycolipids, which are phospholipids with carbohydrate chains attached. So these are the components that make up a cell membrane. What about a cell membrane? Well, now we're going to have a bit of a discussion about the different types of cell membrane, and this is sort of revision from year 11. So first we're going to start over here, where we're looking at our phospholipid molecule. So the main component of the cell membrane is a phospholipid bilayer. So 2 layers of phospholipid molecules. Now, a phospholipid molecule is made up of a phosphate head and a lipid tail. The phosphate head is hydrophilic, so it loves water, and the lipid tail is hydrophobic, so it really fears water. It doesn't want to be near water at all. The cell membrane, truthfully, is held together by nothing but those hydrophilic and hydrophobic natures. So when we get a whole lot of these the phospholipid molecules and we put them in a water environment, they arrange themselves in the fact that all the phosphate heads are attached to water, whereas all our lipid tails are desperately trying to get away from water, so they're being in the inside of those molecules, and we end up with a system like this, where we have all our phosphate heads either facing the extracellular fluid or the intracellular fluid, and then we have all of our lipid tails on the inside where no water or liquid can get to them, so they're being able to stay away from it and making themselves happy because they're hydrophobic. Okay, so the cell membrane is just made up of those hydrophilic and hydrophobic interactions, which makes the membrane form that bilayer that we often talk about. Now, because there's nothing holding those molecules together except for those hydrophilic and hydrophobic nature, that means that the entire membrane itself is fluid. So these molecules are freely able to move around, pinch off, add extra ones. The membrane is fluid. It can move around slightly. We also say it's a fluid mosaic model to cell membrane. It's mosaic because we have all these proteins randomly embedded into the membrane at different points, like a mosaic piece of artwork. In this diagram here, let's explain this a little bit. We have our extracellular fluid, so this is outside the cell. We have our intracellular fluid, which is in the inside of the cell, and then we have the cell membrane separating those two. We have our phospholipid molecules, which have separated themselves into a hydrophilic and hydrophobic nature. We have our cholesterol embedded inside the phospholipid molecules to increase or decrease the fluidity of the animal cell. We have an integral protein that's embedded throughout the entire bilayer and is permanently in that location. We have a peripheral protein which is embedded slightly, can move around, it's not in a fixed location. We have a channel protein which is going to help with transport, and we'll talk about those a little bit later. And we also have a glycoprotein attached. So, what about those other functions of the cell membrane, such as membrane proteins, how they help with hormones? So hormones are a type of molecule that are transported through our blood, and they can only act on cells that have a protein receptor with a complementary shape to that hormone. When that complementary shape meets up with the correct hormone, they bind together, so the receptor and the hormone bind together, and it triggers a cellular change and gene activity inside the cell. So over here we can see we've got our blood, we've got our capillary, we've got our hormone, triangle shaped, and when it finds the cell that it needs to link up with, it exits the circulatory system and it binds to that receptor. It then triggers a whole lot of chemical reactions which are going to cause chemical change within inside the cell. A good example of this is growth stimulators. So we can have a hormone that when it binds, it triggers a whole lot of reactions that strengthens bone and it allows us to grow. Cell recognition, so glycoproteins. These are proteins that act as a cell marker for the cell. There's a very crude diagram over here of that. What they do is they allow the cell to be recognised by other proteins on other cells. So over here we have one cell, and it's got our circular glycoprotein, and we have our receptor molecule in the opposite cell that has a complementary fit. So it comes along, yep, you should be here, let's move along. Why is that important? Well, it allows our body to identify cells that are self or non-self. Anything that's from our own body is protected. Anything that's not from our own body will be attacked by the immune system and taken away. So it's a way of preventing our bodies from becoming infected with different pathogens. Cell adhesion. So proteins help our cells to adhere to different things. And remember, the word adhesion just means sticking to other surfaces. Integral proteins often stick out of the cell membrane, and because of that, that allows them to bind to specific proteins on adjacent cells. They can also attach themselves to the cytoskeleton of their own cell. Remembering that the cytoskeleton is just a framework of tiny protein tubules and filaments that extend throughout the cytosol of the cell. The whole purpose of the cytoskeleton is to attach and move organelles around the cell, as well as to help move cell membrane and chromosomes. So these are the ways in which proteins help with cell adhesion. Another type is our proteins involved with transport, so our channel proteins and pump proteins. Over here we have a channel protein, and this is going to be mostly used in passive transport. So we're talking about diffusion, facilitated diffusion, etc. The main reason for this is we might have some hydrophilic molecules that are physically not able to cross the cell membrane because they're repelled by the hydrophobic tails inside the membrane. Having a channel protein through like this allows them to move through the cell membrane without ever being exposed to those hydrophobic natures, so they're just moving through based on the hydrophilic attractions both inside and outside the cell. We also have carrier proteins and pump proteins. These are a little bit different. These are generally involved in active transport, and we need energy to physically change the shape of the protein to move different molecules in and out of the cell. So you can see at this point the pump protein is open and it's allowing molecules to move into the cell, whereas the molecules on the outside can't actually enter yet because the protein needs to change shape to accept them, and energy will help that protein change shape. Lastly is cholesterol. So cholesterol is a lipid that's only found in mammalian and animal cells. It's not a fat or an oil, it's actually a steroid. Most of it is hydrophobic, so you can see most of it here is actually attached in the hydrophobic tail section of the membrane. But it also has a hydroxyl group, which means this part is hydrophilic, which means it's kind of embedded in the membrane. It's stuck between the hydrophilic and the hydrophobic part. The whole purpose of cholesterol, as I've mentioned a few times, is to just reduce the membrane fluidity and permeability of some solids. So the presence of cholesterol helps change the fluidity of an animal cell, allowing molecules to move in and out with more ease, or to make it more difficult for them to move in and out.